Um, all the papers I'm going to show you guys are available online. So none of this is top secret. So this paper is entitled The Metabolism-Based Inactivation of Penile Nitric Oxide Synthase Activity by Guanabens. So what does that mean? So metabolism basically means that in this context that an enzyme, in this instance, nitric oxide synthase, it can transform a drug. It can metabolize a drug. It can interact with a drug and give rise to another product or transform the drug into something else. So what he found, and I'm going to try to go through this quickly, he found that this drug here in the middle, Guanabens, that's an antihypertensive drug. What it could do to our enzyme in rats was that it could inhibit the activity. I'm not going to get too much into this, the activity portion, but it could cause the levels of the protein to go away. So if you look at this control bar here, that's a control rat. And then the rats were treated with 5 milligrams per kilogram of the drug Guanabens. So you're seeing a loss of the protein. But this drug over here, N-nitroarginine, did not cause the protein to go away. So one drug caused the protein to lose its activity and also to lose the protein itself. The other drug caused the protein to lose its activity, but did not cause the protein to go away. So that's important. So the question is, why is one drug exerting an effect on the protein that another is not? So what's the difference between the interactions of the drugs with the proteins to give the result? What my graduate advisor then did, and his staff of scientists, what they did then was they approached it another way. So we have this data in the animal. So now we need to use a cell model to start to map out and figure out on the cellular level why this particular thing was happening. So they conducted another set of studies here, this time using a cell model. This paper is entitled Guanabens Mediated Inactivation and Enhanced Proteolytic Degradation of Neuronal Nitric Oxide Synthase. Okay, that's a mouthful. But what they wanted to do again was on the cellular level, was they wanted to see what the mechanism for that protein loss was. Okay, so what you can do, I'm going to stop here and go over a couple of techniques. So again, so you have a protein that you're interested in the human and the rat, okay? But you want to study that protein by itself, or you want to study it in a less complex way. Well, one of the things that you can do is you can isolate the DNA from the human or the rat, and you can transfect it. You can insert that DNA into a cell line, okay? And you can ask questions about that protein in this cell line that you can't ask in a rat or in a human. Again, that's called a transfection. So what my graduate advisor and the staff at that time did was they transfected the enzyme into human embryonic kidney cells, and they asked a set of questions. Specifically in the rat, why did the one drug kill the activity and make the protein go away, while the other drug stabilized the levels of the protein but killed the activity? So they conducted these studies here. Once again, you see our drug killed the activity. I'm not going to focus on that at this point. But what they were able to do was they were able to measure the protein in those cell lines. So you have your control, You have your drug here, guanabens, cyclohexamide. That's a protein synthesis inhibitor. You use that with cell lines. What they were able to show was that the drug guanabens caused a loss of the protein here in the cells. So similar to the animal, it was making the protein go away. The same was true for this compound N-methylarginine, which is a substrate analog for this neuronal nitric oxide synthase that was also able to make the protein go away. This drug over here, similar to the animal study, it stabilized the protein to a large degree. So there's something about these two drugs here, the guanabens and the N-methylarginine, that made the protein go away that the N-nitroarginine didn't. So there's a selective mechanism somewhere. They showed similar data here. The guanabens drug made the protein go away. These two drugs didn't. The N-nitroarginine here and the 7-nitroindazole stabilized the protein. So these drugs are doing something unique to the protein. And then this is a time course. Here, you have your control and you're treated, your guanabens treated. And what they saw was that the drug guanabens was able to accelerate the loss of the protein. So this guanabens was doing something to the protein that was making it go away much faster than the control. So what is a clue? What are some clues as to why this was taking place? So if you know that a protein is going away, that suggests that there is a protease involved somewhere. Okay, Rico M.